In our last lesson, we looked at the principles behind how buffers work to resist pH changes in the presence of strong acids and bases. In this lesson, we want to see what factors determine the choice of buffer systems. In this, we will need to draw on our understanding of pH and titration curves and see what determines the capacity of the buffer. In other words, how much strong acid or base may be added without significantly altering pH. Recall that we reached the midpoint of our titration curve, the inflection point, when half of the acid had been titrated or converted to conjugate base. Let's look at the titration curve for the acid dihydrogen phosphate, pictured here. On the left, we have graphed pH as a function of base equivalents added. As strong base is added, some of the acid is converted to conjugate base. At the equivalence point, to the right of our graph, all of the acid has been converted to conjugate base. Now notice the midpoint of the curve, which we know represents the pKa of the acid. Recall that at this pH, the concentrations of the acid and conjugate base are equal. Now notice the flatness of the curve in the area represented by pH values 1 unit higher and 1 unit lower than the pKa. A significant amount of base has been added over this range, and yet the change in pH is minimal. We see the same pattern when we graph pH as a function of relative abundance, as shown in the right graph. These results tell us that buffering capacity is the pKa plus or minus 1 pH unit. You'll want to keep in mind that 1 pH unit represents a tenfold change in hydrogen ion concentration, because pH is on a log 10 scale. We can see then that if the pH changes by 2 units, the concentration has changed 100-fold. Hopefully you can see how this would be outside the buffering capacity range. So then our choice of buffer would be based on finding a buffering component whose pKa value was the closest matched to our target pH. Let me also point out that besides the pKa value, the actual amounts of acid and conjugate base relate to buffering capacity. If we have a higher concentration of each, then there is more buffering or neutralizing capacity. We can summarize the criteria for selecting a buffer as being one that has a suitable pKa and suitable concentration of buffering components. However, there are other considerations, such as not interfering with our experimental assay, having suitable ionic strength, and being one that does not cause our soluble components to precipitate out of solution. Also, we want our buffer to be non-biological in nature. That is to say, it will not interfere with the components of a biological system. Most living systems operate at neutral pH, and most biological components have a narrow pH range at which they function. It becomes vital, therefore, that all cells have some kind of buffering system in order to maintain cell viability.